Hi, thank you so much for inviting me. I'm really happy to be here. Yeah, I'm joining you from North Carolina. It's 9:30 in the morning. It's a very chill day. Yeah, surprisingly cold, but also very sunny. And yes, today I want to show you, since, as Philip said, I just joined the department as an assistant professor. My group doesn't have results yet. So if you invite me in a couple of years, maybe I will show you something that we have done as a group here. But right now I will show you some of the projects that I worked on in my previous career. And yeah, today I want to talk to you about the intersection scene, which is maybe something we don't talk about enough in computational chemistry. So I think I don't have to explain to you that photochemistry is really hard to predict in general. So if you want to ask your computer, hey, what happens if I put these molecules in a flask and I shine light on it, what happens? That's a very hard prediction to make. And the reason for that is that the landscape of these processes is more complicated than for thermal chemistry. So if we start from an initial conformation, we absorb a photon and we are projected on this new excited state, a new potential energy surface. But this time we are not at equilibrium because we just absorbed a photon. So we have an excess of energy. So what happens next depends on many factors, one of them being the velocity that we have since we have this large kinetic energy, but also on the shape of this new potential energy surface. So if there is a minimum, for instance, on the surface, we might get trapped there and then we might fluoresce to go back to the ground state. Or maybe if there is a low barrier or no barrier, we could reach a conical intersection and from then decay back to the initial conformation or form photoproducts. And the things are even more complicated because there are also states with a different spin multiplicity. So all of this is really hard to predict, of course. So how do we usually model computationally photochemistry? Well, the standard technique is nonadiabatic molecular dynamics. And this is a great technique. It can tell us a lot of things. But there are also some drawbacks. First of all, it's an expensive technique. And this is not just because we need to use excited state electronic structure methods, which are inherently costly usually, but also we need to calculate all the couplings between these different electronic states in order to model the population transfers correctly. So an example of the consequences of these difficulties is that it might be hard to catch rare events. So if I have a photoproduct that can get formed rarely, unless I run a lot of different trajectories or I bias somehow my initial conditions, I might not be able to observe this rare photoproduct. But if I need to know in advance what happens, that defies the purpose of predicting, right? Also, sometimes there are some events that are in the realm of photochemistry or photophysics, like fluorescence, that take place on a very long time scale, and nonadiabatic dynamic, being these expensive, might not be able to describe that. So what are the alternatives? Well, there aren't many, but what I want to show you today is what if I explore the intersection scene in a more simplistic way? Can I get the same or a similar amount of information that will tell me what happens when I shine light on my molecule? So, and again, probably this is nothing new for you, but I wanted to bring the point home here. We are dealing with spaces that have so many dimensions. And usually when we represent a photochemical reaction, we draw something like this. We draw two lines that cross at one point. So the two lines represent electronic states. Of course, this is already a 3n minus 6 dimensions surface. So this simple line is just a slice through this multidimensional surface. And then we say, well, these two states cross at the conical intersection. And I want to point out the attention because for me, it was hard to wrap my head around at the beginning. There is no the conical intersection. There isn't one. There is not one isolated point that is the conical intersection, which is this, let's say, double cone that acts like a funnel. But there are infinite conical intersections. So they are part of an intersection seam, which is 3n minus 8 dimensional. So it's less dimensional than the potential energy surface, but it's still a lot of dimensions. So in this example, let's put another dimension on the plot to make things a little bit easier to imagine. If I move across the x axis here, I am lifting the degeneracy, and that's why I have the intersection seam as a 3n minus 8 dimensional space. But if I move across the y-axis, if I follow this red line here, I am not lifting the degeneracy. So I'm still at a conical intersection. So as I mentioned before, once you absorb a photon, you do have this huge amount of extra energy. So technically, you could access the intersection seam in many different points. So we shouldn't talk about the conical intersection because you could encounter so many different ones. So how do we find them? And just to, again, stress this point, if I walk across along the red line, the intersection seam, all of the points composing it are conical intersections. They might bring to different photoproducts, they might have different characteristics, but there are still points of the degeneracy between the two electronic states. So maybe if I am able to find all these different types of conical intersections, I might find some information about what happens after I absorb a photon. So today I want to show you two tales, seam explorations that I've worked on in the past. And the first one deals with, can we automatize the photoreaction discovery? So this project is called the Nonadiabatic Nanoreactor. If you want to learn more, this is the paper that describes the technique in more details. And it's composed of mainly two steps. The first step is a systematic exploration of the intersection seam because we want to find conical intersections. So from the Frank-Condon point, we start what we call a seam dynamics that samples the intersection seam very extensively and finds tons of different conical intersections. And then from that, we reduce their number by identifying the most relevant ones. How is this done? This is actually quite simple. We bias the potential. So the potential contains two extra terms. One is a constraint that is basically pulling my molecule towards the intersection seam so that we don't, quote unquote, waste time exploring other regions of the potential energy surface, but we focus on the intersection seam. And then we have another contribution, which is metadynamics. So we use metadynamics to force our molecule to not get stuck in one local minimum on the intersection seam, but move on and explore as much as possible. From these seam dynamics, we then collect thousands of conical intersections that we can then group together, cluster together based on their geometry. And from the centroid of each cluster, we can find what we call the minimum energy conical intersections, or MECIs. And at this point, you might ask, why the MECIs? Why do we care? If we have this much energy, why don't I consider hitting the intersection seam at a higher energy intersection point? It's a perfectly valid observation. Actually, it's a great observation, but we are assuming here that a minimum energy crossing point is representing a basing on this intersection seam space. So this is an assumption that we're making. The second step in the nonadiabatic nanoreactor is for each of these MECIs, we get the G and H vectors, the gradient difference vector and the intersystem crossing vector. And we use that to build the branching plane. So from this branching plane, we sample some random points that are close to the conical intersection. The idea behind this step is that we have these conical intersections. We want to connect them to the photoproducts that we can actually form if we go through one of them. So in order to do that, we have these points close to the conical intersections, and we let them relax on the ground state or the lower state, and we find these photoproducts. And actually, we also refine the results calculating the barriers and paths connecting them because we want to see if some of them are intermediates or actual final products. And I want to show you what happens when we plug benzene into this machinery. We use benzene because it's a small molecule. It's well known, but its photochemistry is quite interesting because, in fact, when you shine light on it, you do find some very rare photoproducts like newer benzene, benzoline, or fulvene, and they are formed through some interesting intermediates. However, I want to point your attention to the fact that the quantum yield of these minor photoproducts is really, really low. So can the nonadiabatic nanoreactor find them? That's the question that we want to answer. So we use the sim dynamics on benzene, and this is a representation of what happened. These are all, well, some of the conical intersections that we found around 2000. And in order to follow the sim dynamics, we follow these two coordinates, the distance between these two carbon and the pyramidalization of the out-of-plane carbon atom. And as an energy threshold, we use the Frank-Condon energy level. So we start our dynamic here, and you see that the molecule explores this well for a while, and then finally the metadynamics kicks in, and it forces the molecule to move to a different region that it explores for a little while, and then hop again to another well, and so on and so forth. So it keeps exploring different wells. So actually, when we got these 2000 conical intersections, and then we looked for the minimum energy conical intersections, we found 6 of them. These are the representation of them. They are all energetically accessible from the Frank-Condon point, and they are in order of energy. So MECI1 is the lowest in energy. And if you look at them, they look usually similar. You see that in the vast majority of them, there is one carbon atom that comes out of the plane containing the benzene ring. And then mostly the other difference is the orientation of this hydrogen atom here. With an exception, which is MECI number 2 here, because you see that you do break the symmetry in this case, while the others mostly maintain a good symmetry. So, okay, we have found the different types of conical intersections that benzene can use to go back to the ground state. What happens next? We need to do the photoproduct search. So this plot is a bit hard to understand, so I will do my best. So what you're looking at here is the lower portion of the cone. It's color-coded according to its energy. So if you see a region that is more yellow, it means that the cone is tilted a little bit, and that yellow part is closer to you, is higher in energy. All of the circles that are in dark red and are empty, when you start an optimization on the ground state from those points, they lead you to benzene. And this is expected that the majority of them lead to benzene, because remember, when you shine light on benzene, mostly you get benzene back. But then there are these ones. These points are the ones that lead to benzoline. So for MECI number 1, there is a portion of the cone that leads to benzoline. And it's really interesting to see that it looks like a pizza slice. All the points that lead to a different photoproduct are grouped together in space. So it really looks like if you want to form benzoline, you need to access this specific portion of the cone. This is interesting. Is this a common feature, though? Well, it turns out, at least in benzene, this is exactly true. I'm showing you here only the 3 MECIs that are the most interesting ones. And you do see that in every different MECI, you do access a different photoproduct. But the points that lead you there are grouped together in the pizza slice configuration. So this seems like it's a common feature. So MECI forms benzoline. MECI number 2 form transbenzene, which is an intermediate in the formation of duobenzene. And MECI number 4 forms prefulvene, which can form fulvene as well. So even from this simple machinery, we are able to find all the photoproducts or the intermediate to the photoproducts. Okay, can we actually estimate the quantum yields though? Or do we know how much of each we form? And here I'm showing you something that we haven't published yet. It's a very simple model. We said, can we get an estimation of these quantum yields in the simplest way possible? So if I calculate the probability of forming the photoproduct J, I can do it as the sum over all the conical intersections I of the probability of reaching that conical intersection I, and from there, the probability of forming the photoproduct J. So this one is kind of hard to estimate since we don't have any dynamic data. The simplest thing you can think of is to use some sort of partition function here where you have this weird equilibrium between these conical intersections. And if you want to estimate the energy that you have, it would be the difference between the energy at the Frank Condon point and the energy you would have on the minimum on the excited state. It's a very crude model. This other part, well, we could use the data that we just produced. So we could use the area of the pizza slice. So here we're running very simple calculations. And again, there are many approximations behind it. We are considering as if the MECIs were in equilibrium with each other, which is definitely not true. We are considering that each part of the cone here is equally accessible, which is to be proven at least. And also this temperature that we are calculating here is estimated in a slightly weird way. So does this simple method works? Well, at least for benzene, it works surprisingly well, even too well. You can see that the theoretical quantum yield and the experimental ones are very, very close together. So you can now think that you could improve on this model by lifting some of these approximations one by one and see which one are more relevant. But the bottom line is that as of today, the Nonadiabatic Nanoreactor can indeed find photoproducts for small molecules in gas phase. So it can tell you what you are forming and roughly how much of it. So this was the first tale that I wanted to tell you. And now we switch to a second one where we are actually talking about proteins. Fluorescent proteins is the topic that we are dealing with here. And these are great tools. These that you're seeing here is the green fluorescent protein. And the reason why they are so important in science is because they are used in imaging, in biological imaging, especially in vivo imaging. And you can really produce amazing pictures with a great crispness to study some biological processes. Mostly, in this talk, we will focus on the red fluorescent proteins because they have been, the original one has been engineered, so you have basically a rainbow of color that you can choose from. But the red ones are really interesting. They are really interesting because, first of all, red light has a great light penetration. So if you want to image deep tissues in vivo organisms, that's what you want. You want a red light or near infrared, even better. It has also other good things. It's not too damaging for the cells. It gives you crisper images. However, there is a big con. The con is that usually when you engineer this protein to make it emit in the red light, if somehow the protein becomes not very bright experimentally, which makes it really hard to use. So wouldn't it be good if we could know how to improve on a protein computationally to make so that it's both red light, red shifted and bright? So first of all, we need to understand why are some of them so low fluorescent quantum yield? How do we model fluorescence on a computational framework? Well, if a molecule is fluorescent, it means that there is an excited state minimum at some point. So the molecule gets trapped there for long enough that the only option it has is to emit a photon to go back. So it could either be that the conic intersection is higher in energy than the minimum, or maybe it doesn't need to be lower, but there is a barrier to get there. So on the contrary, if a molecule doesn't have a very high fluorescent quantum yield, it might be that the minimum it gets stuck to doesn't have an oscillator strength that is really high, or somehow the intersection seems accessible. It might be very close in energy, or if there is a barrier, it's a low barrier. We also need to remember what we just talked about. There are different types of conical intersections. So maybe some types are accessible and some others are not. And this is specifically relevant in this case because the fluorescent protein chromophore can twist around two different dihedrals after the photoabsorption. So if you twist in different ways, you might reach different conical intersections. So actually asking how bright is this molecule means, in computational terms, to estimate how accessible is the intersection scene. And it strongly depends on the shape of this new potential energy surface. So in this project, we compared two red fluorescent proteins called M scarlet and M rouge. They are very similar from a sequence point of view. There are just a few point mutations here and there. And in fact, the absorption and emission are also quite close together, but the fluorescent quantum yield is very different. M scarlet is the bright one, and M rouge is basically dark. So, can I just observe the intersection seam of these two proteins and find the reason for this discrepancy there? So what we did computationally, we used classical MD to sample conformations. So we got then 10 snapshots per molecule, per protein. We did a QMMM optimization on the ground state to find the ground state minima. And we did optimizations on the excited state to find the excited state minima. Then what we did was to twist the chromophore in different ways to find all possible types of conic intersections. And finally, we calculated the path connecting the excited state minima and the conic intersections to see if there is a barrier, what kind of barrier is there, and so on. And as a detail, these calculations were really expensive because we used a QM region that was between 70 and 100 atoms for the optimizations, but then all the energies were refined with a much larger quantum region and CAS-SCF. So this was pretty intensive. So what did we find? So again, I remind you that this chromophore can twist around the dihedral I and the dihedral P. So you can twist, of course, clockwise or counterclockwise. So you can, in principle, find different types of conic intersections. So we found the type I+, rotating around I+ in a clockwise direction, the P-, the I-. And then if you observe here, you will not see a P+ type. And this is because the protein environment is chiral. It prevents some types of rotations. So another type that we found instead is what we call the hula twist, where the two rings are basically remaining coplanar, but the bridge here is out of plane, it's rotating. So 4 different types of conic intersections. Now, this is the important plot here. These are representing the energy of these conical intersections with respect to the Frank-Condon point. And the black dots are the average and the standard deviations as error bars. So when we look at M scarlet, we see that basically all of the conical intersections are higher in energy than the Frank-Condon point. So this explains why M scarlet is most likely bright, is very bright. It has a high fluorescent quantum yield because it's simply very difficult for this molecule to get from the Frank-Condon to the intersection scene. On the other side, when we look at M rouge, and you will see M rouge A and B because there are 2 conformations for this protein, and we simulated both, but they behave in a very similar way. You can see that some conical intersections at this point do become accessible. So in principle, it could be possible for the molecule to go from the Frank Condon to the conical intersection here, unless there is a huge barrier in between. So this might explain why M Rouge is so dim. It's simply very easy for it to get to the conical intersections. And I am telling you, there is no barrier. So since these two proteins are so similar to each other, how can we find out what is the mutation that is responsible for this huge discrepancy? So we did an analysis calculating energy differences between the energy of the Frank Condon point and the energy of the MSCI by stripping the chromophore from the snapshot in the protein and putting it in gas phase. This is what we call the bare chromophore. It's just the chromophore in that specific geometry without anything around it. And then we redo the calculation, but with putting one side chain of one amino acid at the time close to it and recalculating this energy barrier. Is it increasing? Is it decreasing? This will tell us what kind of impact has each amino acid. And the results here are interesting. So first of all, you can see that some amino acids that are conserved have a specific function. For instance, this arginine 88 or 93, which is conserved, it mainly stabilizes the MSCI I plus type in both proteins. And similarly, arginine 63 or lysine 68 have a similar effect, but on I plus. So it stabilizes the I plus type. So these two is just a confirmation that this analysis makes sense because it's consistent. So how do we find the other residue? We observed that there is a specific mutation, arginine 190 to threonine 195, and this is destabilizing all the possible types of conical intersections, but it is destabilizing them in different ways. So this one specifically is the one that is making the MSCI I plus kind of inaccessible in M scarlet, but partially accessible in M Rouge. And it makes sense that this is an important mutation because you're going from a positive, bulky amino acid to one that is neutral and much smaller. So this has a strong impact on the photochemistry of this protein. So in conclusion, I hope I showed you some interesting things about the intersection seam. In this case here, we did just, let's say, a static exploration on the intersection seam, and we could find that, yeah, the intersection seam is not as accessible in the right protein as in the dim protein. And we could also find what amino acids are responsible for it. And we could use this information in the future to modify the protein to make it behave like we want. And before, I also showed you that just a systematic exploration of the seam can tell you what can happen to a molecule in gas phase when you shine light on it. So why is this important? Well, we do have, as computational chemists, the dream of being able to just go to the lab and design a new molecule and save a lot of time and effort to experimental people. And this is, if you're asking, this is what Dali thinks this future could look like. And definitely, if we are able to be more predictive, this is what we could achieve in the future, being able to have a positive feedback with computational and experimental people where we inform each other's work. So as I said, this is mostly work that has been done during my postdoc at Stanford. Many people contributed to these projects in different ways, and these are some of the institutions that funded us. And yeah, now I am a new assistant professor at North Carolina, Chapel Hill. This is my group website. I also want to take this opportunity to invite people to apply to our graduate student program, because we would really like to start some exchange with European students. So if you're thinking about an American adventure, or if you have students thinking about it, please share the news. And with this, I thank you, and I will take questions.